Hi, I'm Michelle. We are from Group 8. Today we are going to present about the transgenic plants with enhanced nutritional traits. Transgenic plant is a plant that has a modified genome, which is done by the genetic engineering techniques through the insertion of transgene that originated from other species. So the transgenic plant acquires a new beneficial trait that does not occur naturally. For example, increasing of yield, having tolerance towards various environmental conditions such as extreme temperature. The transgenic plant even have resistance to pest attack and disease. In this presentation, we are focused on the second generation of transgenic plants, which are produced with the aim of improving their nutritional value. Transgenic plants with enhanced nutritional value can be called as biofortified crops. There are nearly 2 million of world population suffering from malnutrition due to insufficient intake of essential micronutrients such as vitamin A in their diet. So, the production of biofortified staple crops can improve the nutritional status in developing countries where their people are less accessible to nutritious food. The animal feed crops also can be engineered to have high amount of limiting amino acid for improving the growth of the animal, which then reduce the cost spending on supplementation and also reduce the nitrogen excretion into the environment.
Hi, I'm Paying. Now I'm going to talk about the methods to produce transgenic plant. First of all, the agrobacterial-mediated transformation (AMT) is used. The genes of interest is first isolated, and the gene is later inserted into the TI plasmid of agrobacterial tumorphages to recombinant DNA technology. After that, the tDNA is introduced into the agrobacteria, and the bacteria is used to infect the plant cell so that the tDNA can be integrated into the plant genome. The strength of using the agrobacterial-mediated transformation is it's easy to use, inexpensive, low transgene copy number, minimum DNA rearrangements, and high stability of transfer gene, while the weakness is several athlete monocoque copes, legumes, and woody plants are resistant to agrobacterial tumorphages. The second method used is microprojectile bombardment. First of all, the gene of interest is coated with spherical gold or tungsten particle, and then it is accelerated to high speed with particle gun so that the gene of interest can penetrate into the targeted plant tissues. The strength of using this method is wide range of plants and tissues can be used, and it is an excellent and efficient system for transient expression, while the weakness of this method include multiple copy DNA insertions and fetchments and loss of molecular integrity of the DNA. There are also traditional transformation methods that can be used to produce the transgenic plant, such as photopath transformation by electroporation, microinjections, or polyethylene glycol fusion. The weakness is they are time-consuming and they have only low efficiency.
Hello, my name is Hu Xiong Ek, and today I'm going to speak about the transgenic tomato. First ever engineered tomato is the Flasaf, which is the Flasaf is also called as flavor saver. It's developed through using the antisense RNA to regulate the p-tom6 gene, which subsequently control the productions of PG enzymes. And PG enzyme usually is responsible for the softening of the fruits, typically when those tomato fruits are ripening. Hence, when we consume Flasaf, we have higher in pectin content and provide better content of fiber to ourselves. So moving on to the next engineered tomato, which is Elsa Cray tomato. It has increase of beta carotenoid content up to 45%, while its peer is Elsa Cray tomato, which it has twofold increment in productions of fruit carotenoids, phytoin, lycopene, beta-carotene, and lutein. This beta-carotenoid, beta-carotene, lutein, when we consume it in our human body, we subsequently convert it into vitamin A's for hence we have better eyes and better skins. Moving on to the engineered tomato, one of the example is it also has significant increase in level of folate, which is essential vitamin B's. And vine ripe tomato has 25-fold more folate than a normal tomato. Hence, it provides enough folate level for adult less than 1 standard servings. That's all from me.
Next, we will discuss the nutritional enhancement in transgenic rice. As we know, almost half of the world's population depend on rice as their staple food. Hence, nutritional enhancement in rice could reduce nutrition deficiency in low-income population. One classic example for rice is the golden rice, which have been described by the Time magazine as a rice that could save billions of children. In 2013, 29% of children aged between 6 and 59 months in low and middle income countries were vitamin A deficient, and it causes blindness and also child mortality. So in 2000, Ye et al. Developed a provitamin A enriched rice with introduced B-carotenoid biosynthesis pathway in rice that significantly enhanced the vitamin A density in the rice. Furthermore, other nutritional improvements in rice include mineral enrichment such as increased iron content, increased amino acid levels as some seed proteins of rice are deficient in some essential amino acids, and improved oil quality such as increased alpha-linolenic acid levels.
Next, transgenic plant also done in a potato. As you can see here, a SE potato has about 14-fold increase of the beta-carotene compared to the wild type 1. They're using the agrobacterium-mediated transformation to silence lycopene cyclase gene that affect the beta-carotene synthesis. Beta-carotene is the precursor of the vitamin A. Besides, the transgenic potato has the calcium content that have been improved about 50 to 65% compared to the control one. They are also using the same method, which is the agrobacterium-mediated transformation method, which is in this case, they want to increase the capacity of the calcium ion transporter. Based on the previous study also states that these potato tubers shows no adverse effect, which is in the term of there is no accumulation of the heavy metal, which is manganese, and also there is no morphology changes in these potato tubers.
Hello, I'm Wai-Ling and I'll be talking about the controversy of transgenic plants. The use of transgenic plants is still highly debated among the community. The division in opinions on transgenic plants is due to various reasons. Consumers are skeptical of the safety of the consumption of transgenic plants, as the side effects of prolonged consumption are unknown. The consumption of transgenic plants may bring potential health risks to humans, as the transgenic plants can be a potential allergen, toxic to humans, and causes antibiotic resistance in humans. In terms of environmental effects, the transgenic plants can become invasive species, causing characteristic alteration of the surrounding flora due to genetic contamination and the emergence of superpests. Transgenic proteins can cause unintentional toxicity to other plants and organisms. Possible horizontal gene transfer is also a concern over the use of transgenic plants, as it may produce antibiotic-resistant pathogenic microbes and also superweeds. Many people view transgenic plants as unethical, as the insertion of gene into a living organism is unnatural and seen to be opposing many religious beliefs. To sum it up, transgenic plants such as tomato, potato, and rice have shown enhanced nutritional traits such as increase in vitamins and mineral contents that benefit human health. However, the use of transgenic plants is still controversial and highly debated among scientists and the community. Transgenic plants is indeed a powerful tool in solving the food security issue faced. However, more studies and tests should be carried out on the transgenic plants to ensure its safety towards humans, animals, and the environment. Thank you.